Okay, we'll wait another couple of minutes to get started. In the meantime, you guys have any questions? No one? Okay, shall we start, guys? All right, so if you haven't seen the announcement, today is the last lecture, so no lecture next week. Your final exam is fairly early, which is December 5th, okay? So I'll give you more information about the exam at the end, but basically it's going to cover lecture 5 onwards up to today's lecture, okay? There is a quiz next week, so please make sure you guys complete your quiz next week as well. And lastly, for your exam viewing, I'm going to post a Microsoft booking form for you guys so you can sign up for the exam viewing slots. It'll be sometime next week, okay? So you can come see your exams if you wanna see where you made your mistakes, okay? All right. And lastly, I think your assignment as well, it should be graded by the end of next week, but you'll definitely get it back before your final exam, okay? All right, any questions before we start? All right, so far we have seen all sorts of different viruses, and this time we are going to be talking about how to prevent those kind of infections. So we're going to be looking at antiviral vaccines as well as how we can actually design drugs or what are some of the antiviral drugs that are commonly used, okay? So we'll go with that. So starting with vaccine history, a little bit of antiviral vaccine history. So remember that this is the most effective means to prevent viral infections, okay? So rather than treatment itself, vaccination works better in a lot of cases. So this is some examples here, and you can see how the numbers have changed. So look at this, for example. all 100%. If you look at the next three, they are almost close to 100% as well. So these vaccines, they work very, very well. So one thing to keep in mind, though, is if a vaccine does have serious adverse effects, it needs to be investigated. But these adverse effects are usually very, very rare because these vaccines do undergo rigorous testing as well. And if a vaccine is not 100% safe or effective, then it raises ethical concerns. Like, do you really want to give that to someone when it has more harm than advantage? Okay? So this is something to keep in mind, that yes, vaccines can have side effects, but most of those side effects are manageable. And we saw some early vaccination techniques. So remember, they were crude methods, but they worked fairly well. So we saw with smallpox where they used powder smallpox scabs to actually immunize the patients. Okay? Then you had immunization with pus from a recovering smallpox patient. Okay? And then they started using things like, for example, air-dried rabbit spinal cord for vaccine against rabies. Okay? So as we develop new and new technology, vaccines were changing as well. So these crude vaccines from natural products work well, but because of the technological limitations, there were some serious side effects. Okay? So it was very difficult for the general public to be convinced to actually get vaccinated, because this was one of the problems was there were ongoing epidemics, and some of them had really visible effects, like we saw, for example, with smallpox. So this was often the reason people would get vaccination, because the alternative is much worse. Okay? And if we continue along the development pathway, we see that we started making primary cell cultures and immortalized cell lines, and that actually accelerated the development of vaccines in the 50s and the 60s. So there were three major leaps that were required. One is how to carry out serial passaging of viruses in humans or in animals in general. So what's serial passaging again? What's serial passaging? Yeah. Like multiple times in succession. Okay? So you carry out serial passaging, and generally you carry out dilutions as well around that time. And the viruses, we eventually started growing some of these viruses in sterile conditions as well as embryonated chicken eggs. So basically you could purify the vaccine from the chicken eggs, or purify the virus, from the chicken eggs. And we were able to grow some of these viruses in vitro. So we were able to grow these in culture. And again, we saw some advances in this case where we were able to pass, for example, rabies from rabbit to rabbit. So it was shown that it also involves a virus. Okay? Then, when Petri dishes were developed, we were able to start with cell culturing and use embryonated eggs to culture viruses as well. Okay? And then from there on, we started studying those, and the first immortalized cell line, the HeLa cells, they were started to getting to use to study viruses and understand their various mechanisms. Okay? So there are a lot of different technological leaps that had to actually take place for the vaccines to be at a state we are at currently. So let's look at an example. In this example, we see that if we try to make vaccine against avian influenza, it's been a little bit problematic. So this, because one of the major panzootic strain, which is H5N1, that happened in 97 all the way to 2009, okay? Panzootic means there's a widespread increase in infectious disease in animals over a very large geographic range. So this is basically an epidemic in animals. You guys must have heard the recent news that came out of Vancouver, or came out of BC, I should say. Anyone know what that is? Related to bird flu? No? You guys didn't see the news? So there was this farm where there was an outbreak of bird flu in ostriches. So they actually had to kill all those ostriches, okay? So this kind of stuff can happen. It's actually happening more and more frequently, okay? So for example, this kind of strain is highly pathogenic in birds, but it rarely causes infection in humans. But the problem with this is it's only a matter of time before it actually jumps from birds to humans. And if the virus mutates enough, it could lead to a major pandemic in humans, and it can have mortality rates up to about 70%. So it could be as deadly as things like we saw earlier, like Ebola. Okay, and if you haven't, did you guys see about the outbreak in Guyana, I think? No, not Guyana, somewhere else. Can't remember where exactly. There's an outbreak of Marburg virus since last week, okay? So this can become a major issue worldwide again. Now, there are some serious technical problems for vaccine production for this kind of example where the virus recombines and mutates quite often, because the virus itself in this case was so pathogenic that you couldn't actually develop enough virus, okay? Because the chick embryos were killed before sufficient vaccine virus was even produced. So that became a major issue, but then we had new techniques that we could use to actually produce these vaccines. So what we are going to do is we are going to look at some of the classical categories of vaccines, and then we get to the mRNA vaccines that are more common now, or becoming more common now. So there are two different categories for classical vaccines. And one is the live wild-type viruses. So in this case, you basically just take live virus and you infect a person to give them immunity against that virus. Very simple method, works really well. And one issue with this is that the viruses will replicate poorly in a non-natural host, okay? But they do share immunogenic determinants. So this is the regions of the virus that have the ability to induce an immune response with related targeted viruses. So for example, you can use vaccinia virus to vaccinate against smallpox, okay? That's one. Then you have live attenuated viruses. These are generally the most effective way of getting immunity against a particular virus. So you carry out serial passaging, and you cultivate the virus in vitro, and that actually reduces the pathogenicity, okay? And I'll explain how that does that in a minute. So you can either use the live wild-type virus, or you can use the live attenuated virus. So what that means is the virus is not as effective or not as pathogenic, okay? So this becomes an important part here, the serial passaging. So let's see how it works. So the virus itself is grown in vitro, okay? And over time, as we continue to pass that virus into the next set of cells, it will carry mutations, or it will become mutated, okay? And overall, after several passages, it may become less pathogenic. As a result, it can replicate in humans, and it can give immune protection, and it may or may not induce little or no or little disease in those patients. So how does that work? So here's your standard virus. And let's say you infect the cells with MOI of 10. Okay? You let the virus grow, and you isolate the virus from these cells, and again, infect another next set of cells with the same MOI. And in these populations, some virus will be defective. As we have seen, we do get defective virions as the infection progresses. All right, you continue doing that, and what will end up happening is you'll end up with more and more defective viruses in this case. Okay? And this is what you can use to develop vaccines. And some of these techniques, they actually modify the virus such that they don't grow very well at less than 37 degrees. So these are cold-adapted vaccines. So one example is a cold-adapted influenza A virus. And this cannot actually replicate in the lung tissue, but it can in the nasal mucosa. Okay? And from there on, it induces an immune response. Okay? So in this case, it won't cause an infection inside the lungs, but it will be present in the nasal passages, and from there, it will induce an immune response. Okay? Now, within that, remember, you can also do inactivated viruses. So we saw live viruses and live attenuated viruses. Okay? Next is inactivated viruses, where the virus is grown, it's purified, and it's inactivated using various methods. So one method is using chemicals. So for example, using formaldehyde or even organic solvents, it will render the virus inactivated. Okay? You can also do this with high temperatures. What do high temperatures do generally to a protein? What would high temperatures do to a protein? It will denature part of the protein. Yes? So you carry this out at a higher temperature, and that again inactivates the virus. You still get an immune response, but you don't get an infection. Okay? So some of the examples are here. So this is live wild-type vaccine, so cowpox or vaccinia virus. Your attenuated vaccines are here. Inactivated viruses are, for example, flu viruses or hepatitis vaccines, etc. Okay? Next you have is subunit vaccines. And this basically has a purified viral proteins that are immunogenic, but they do not have a genome. So basically, you purify viral proteins, you process them, and then you inject them or give them to the patients. They still induce an immune response, but there is no genome, so they can't cause an infection. These are actually relatively easier and cheaper to produce using things like cloning. Okay, so you can actually clone this into a vector, put it into the cells, and isolate that protein. You can do that in, for example, eukaryotic cells. You can do that in bacteria. Okay? And lastly, you have the virus-like particles, or VLPs, and they are generally more effective than single protein. So again, this is just the protein shell, but no genome. So as a result, it's not infectious either. So HPV vaccine, for example, is virus-like particles. Okay? Then you have your live vaccine. Live vaccine is where the virus actually replicates in the host cell and produces progeny virions. Okay? And in this case, both your progeny virus and the viral proteins that are made within the infected cell, these are recognized by the immune system as well. This generates the cytotoxic T lymphocytes and specific antibodies against that virus. So this actually kills the infected cell types as well as neutralizes the virus. So here's an example. Here's your live vaccine. You inoculate a set of cells, or you inoculate the host cells, and these viruses will replicate. From there on, once they are released, they are recognized by the immune system. You produce antibodies. You also produce cytotoxic T cells. And both your infected cells and your virus particles, they are neutralized. Okay, so in this case, you kill the virus as well as the infected cells. So this can work for other vaccines as well. So here's an inactivated vaccine, same thing. You induce an immune response, etc. And here's a subunit vaccine. Okay, because both these types of vaccines, they actually do not replicate in the host, but they're still recognized by the host immune system. Now, one thing to add on here is that you can add material that makes it better, because generally the vaccine on its own can be less efficacious and is often administered with what are called adjuvants. Adjuvants are non-antigenic compounds that enhance immune response in your body. Okay? So you take an antigen and an adjuvant, and this will allow you to have a better immunity. But in some cases, you may require boosters. So there are some vaccines that require booster shots. Why? Why is that? For example, when COVID vaccine was going around, we had to get booster shots. Why?
Maybe due to chronic infection.
Sorry?
Maybe due to chronic infection.
Not chronic infection, no. Yeah?
Well, viruses like COVID-19 may undergo mutations, so we got new variants and we got protect ourselves.
Okay, so you could get vaccinated for new variants. That's one. But remember, our immunity goes down over time as well. For example, you have to get a tetanus shot every 10 years. Tetanus is caused by bacteria. Yes? But you still have to get the shot every few years. That's because our immunity eventually goes down. So you have to prime your immune system again with those vaccines. Okay? And like we saw earlier, the wild type and the attenuated live vaccines, they result in an immune response that comes the closest to the natural infection. And generally speaking, it gives long-lived immunity, okay? So more than 10 years. Delivery is simple. Some can be taken orally or nasally. So a lot of the times for kids, especially like if it's not being injected, you give them a spray in their nostrils, okay, in the nasal passages, okay? Or you just give it to them orally. But the problem with this is they are expensive and they are unstable. You're working with live viruses. Even if it's attenuated, it's still a live virus. So what that means is that you need a cold supply chain. So once the manufacturer makes the vaccine, it has to stay cold at all times because temperature fluctuations will denature the virus, okay, especially if it gets hot, okay? So this cold supply line has to be maintained from the manufacturer all the way until it's administered to the patient, okay? So these kind of vaccines are sensitive to heat as well as freezing. So you can't freeze them, you can't heat them. You have to keep them at a right temperature, okay? And in some cases, there may be an inactivation by maternal antibodies when they are administered in early childhood. Because remember, antibodies do pass on from mother to the child. So some of that response may inactivate some of these or part of these vaccines. But one good thing about this is they rarely cause serious illness or death. Yeah?
Is that why you can't just like vaccinate the newborn?
So that's part of the reason they don't vaccinate the newborn, because for now they are getting their immunity from their mother, okay, especially if they're breastfeeding, for example, okay? Now, there are some inactivated viruses, inactivated vaccines as well, but in this case, they are whole virus vaccines. And they can actually, you can generate an immune response to a number of different viral antigens. Okay, so you can actually use a mixture of antigens in these kind of vaccines. They're less sensitive to storage issues. So the virus is already inactivated, so it doesn't really matter. You still have to store them under right conditions, but they're not as sensitive as live vaccines. Also safer for immunocompromised individuals. They are a bit expensive to produce, and they do require manipulation of virulent pathogens. So you have to be very careful when you're making these viruses or making these vaccines, okay? But one thing to note is that they do not induce a robust immune response, because that's why it requires things like multiple doses, boosters, etc. Why is that? Why is the immune response not as strong in this case? Yeah.
Because replication doesn't occur and the virus doesn't get inside the cell.
Okay, so the virus is actually not replicating inside the cell. Your body is just creating an immune response to whatever dose of inactivated virus you got. Okay? So the virus itself is not replicating. You still do induce an immune response, but it's not as strong. Okay? And these have to be injected, and they do generally require an adjuvant to get a strong immune response. Okay? Now the next is the subunit vaccines. These are the simplest and the least expensive vaccines to be made. They are very stable and very safe in immunocompromised patients as well. Because one major advantage of this is that you don't deliver any pathogenic viral proteins causing adverse effects. So it avoids the delivery of irrelevant and potentially immunopathogenic viral proteins. Again, just like other vaccines, you do need multiple doses and boosters, and they typically do require an adjuvant to get a better immune response. Okay? Then you have the newer categories. So those are the classic categories of the vaccines. Now these are the newer categories. So one is the chimeric vaccines, where chimera basically means it has more than one genotype. So what this has is live but genetically modified organisms. So basically, you take a non-pathogenic organism, okay, and then let it express viral proteins of interest. Okay, so you can also use a non-pathogenic virus and express those viral proteins. Where did we see that? Last week. Or maybe the week before. Anyone remember? No? Remember we were talking about pseudotypes? Who remembers pseudotypes? Somewhat rings a bell? Okay. All right, pseudotypes are, remember, unrelated viruses that express a viral protein that you want to study. So in this case, it's similar. You can make a non-pathogenic organism, doesn't have to be a virus, and you can express the viral proteins of interest, okay? And it can also deliver antigens to desired locations. So you can actually target specific locations with these kind of vaccines. So let's say in the dendritic cells or in gut mucosa or wherever you might want to target this. And the newer one is the RNA and DNA vaccines. And this is obviously based on mRNA and or DNA plasmids that encode one or more viral proteins that can be expressed in host cell. And then your body will have an immune response against that because it's a foreign protein. Okay? So you have a transient expression of these proteins. They go to the cell surface, or the cell itself will trigger the immune response, and from there on, your body will develop immunity to this. So for example, the COVID-19 vaccines from Pfizer and Moderna, those are mRNA vaccines. Okay? Now, in addition to this, there's also nanoparticle vaccines, and this includes the virus-like particles. Okay? Because what you do is you make synthetic nanoparticles, so liposomes. Okay, you basically very, very tiny nano-sized lipid vesicles which display multiple copies of your viral antigens, and then you can combine it with adjuvants. So Novavax vaccine was using the synthetic nanoparticles. And the newer thing is basically you can actually package things like mRNA into these nanoparticles and target it to specific cell types. Okay? How would you target that? How would you do that? Let's assume we are working with COVID-19 vaccine. How would I target it to the cells that are normally affected by the virus? Okay, so you use, for example, the viral surface protein. So for COVID-19 vaccine, spike protein was used. Okay, so you can actually package the mRNA in the lipid nanoparticles. They go inside the cell, and from there on, you get a productive infection where the cell will start making the spike protein. And from there on, your body will develop the immunity. Okay? And a lot of these recombinant vaccines, they tend to use genomic fragments to encode immunogenic viral proteins. So they are generally engineered into a non-pathogenic virus. So for example, you can do that in a non-pathogenic virus or even bacteria. Okay, lactobacillus is commonly used. Or you can also do this into a plasmid, where once inside the host, you'll start expressing that protein, and as a result, you'll get an immune response. You can use this to make virus-like particles, nanoparticles, and even multivalent peptide vaccines. Normally, what you see is a peptide will have a few epitopes that the antibodies can recognize. In this case, it will have hundreds of epitopes that can be recognized. Okay? So here's a chimeric virus. So this is a virus that's been genetically modified. You let it infect the host cells, and then you're going to make all these extra virus particles. Okay? This will get recognized by the immune system. You produce your CTLs as well as your antibodies that will bind the virus. Here's a DNA vaccine. You basically get that DNA inside the cells. The DNA has the genes for the viral immunogenic proteins. And again, these produced proteins will induce an immune response. Then you have your virus-like particles as well as nanoparticles. Okay? So you can actually add additional genes. So for example, antigens against cellular pathogen recognition receptors that can be incorporated to get a stronger immune response as well. So you can target the receptors as well. So let's look at an example. In this example, we are looking at the COVID-19 vaccines. So AstraZeneca vaccine was actually a virus vector DNA vaccine. Pfizer and Moderna were mRNA vaccines. Okay? So you can see that in the figure as well. Here's your replication defective virus vector DNA. So you basically added the DNA to take out all the replication genes, but you add your specific target in this DNA. This will go infect the cells. Once it makes the proteins, it will induce an immune response. Okay? Same thing here, mRNA as well. You have an mRNA vaccine into a lipid nanoparticle. This will enter the cells, make the protein, and induce an immune response as well. Okay? So there's multiple ways to actually make vaccines. And from what we have seen in the last few years, these are becoming more and more common. They're actually working on mRNA vaccines against various types of cancers as well. Okay? And just to show you guys, these are all the different COVID-19 vaccines. This is where it was developed and what they use. So you guys can go through this if you want to understand how this works. But again, this is not something I expect you to memorize. Okay? Like for example, this one right here uses whole virion. Okay? Here, spike protein. Okay? Etc. And just to show you guys, here's the Moderna Pfizer vaccine. So what they did was they made this kind of mRNA construct. So it has a poly A tail, it has a cap, untranslated regions, and that's the gene for the spike protein. Okay? Now, these are all packaged into a lipid nanoparticle, and some of these, they actually have some modifications. For example, in this case, it has a pseudouridine, which makes the vaccine a bit more stable. Okay? Novavax is a subunit vaccine. Again, it has the spike protein, but within the spike protein, there are multiple different mutations. So for example, R682Q, what does that mean? Those are mutations. So what does that mean? Yeah. Okay. Sorry, say that again. Okay, so that basically indicates what the mutation is specifically. So R682Q means arginine at position 682 has been replaced by glutamine. Q stands for glutamine. Okay. This is lysine at position 986 replaced by proline, for example. Okay. And again, this is packaged into a lipid nanoparticle through a baculovirus genome. Your adenovirus vaccine, which was the AstraZeneca vaccine, is basically what they did was they took the spike protein gene and cloned it into a specific adenovirus genome. In this case, it was a chimpanzee adenovirus genome. Okay. Janssen did the same thing, the Johnson & Johnson vaccine as well. Spike protein with mutations, and then it was assembled into, or it was cloned into an adenovirus genome. So they also deleted some of the pathogenic regions of this, and as a result, the whole thing was packaged into the genome, or into the genome of this virus. And then when you got the vaccine, this virus went and infected your cells. That's where you started making those proteins and developed that immunity. So far what we have seen, the clinical data, these were the most effective vaccines. Okay? Any questions so far? Okay. Now, for the antiviral vaccines, there are two major mechanisms that you get the immune response from. One is the cellular immune response that mediates the recovery from a majority of virus illnesses. In this case, you kill the infected cells through your cytotoxic lymphocytes, and this is the main mechanism to eliminate infection from the body. Okay? You also get neutralizing antibodies. And these neutralizing antibodies can actually confer protection to most of the viral pathogens. They limit infection and reinfection, and they also provide cross protection against viruses that are similar, okay? So let's say another virus emerges and it's very similar to the COVID-19. It has a protein that's very similar to the COVID-19 spike protein. So as a result, you will have some immunity to that virus because the structures might be similar. Your T cells and B cells, the memory T cells and B cells, they support the long-term production of the neutralizing antibodies. And you also get passive antibody transfer from mother to infants to actually protect the babies, okay? So antibodies protect against the infection, and the cellular immunity itself helps clear the infection or helps with the recovery from an infection. Because the generation of your antibodies is not always sufficient to control some of these viruses. So for example, viruses that establish latent infections, so things like herpes virus. Viruses that have multiple different types of variants or serotypes, okay? And they have limited cross-neutralization. You can also have issues with rapidly mutating viruses, so influenza A virus, okay? So that's where you get your flu shot. And lastly, viruses that actually establish chronic infections, so things like HIV. But one new development is that the HIV genome can actually be edited out of our cells using CRISPR-Cas9. It's not mainstream yet. It is still in clinical trials. So there are other approaches by enhancing cellular immunity, for example, which does not really require antibodies. But one thing that's clear from all this is that there's no current universal fit for the development of antiviral vaccines. For each of the virus, you'll have to use specific type of vaccine, okay? So there's nothing that, okay, if we make this type of vaccine, it's going to work for all viruses. That doesn't happen, okay? And there's somewhere in the world, there's always an urgent need for vaccines against viruses that cause significant illnesses. So RSV, HIV, dengue virus, etc. Okay? Now, next thing is we're going to talk about is the basically adjuvants. And remember, adjuvants, they play an important role in vaccination, and new adjuvants are under constant development, okay, to get us a better immune response. So you can add this to boost immune response, and as a result, you produce more antibodies and longer-lasting immunities. And as a result, you minimize the dose of the antigen that's actually needed. It also stimulates secretion of cytokines that favor the production of antibodies as well, okay? So adjuvants can trigger that response in our body, and they will allow to produce antibodies in larger quantities. Now, generally speaking, they are not required for live attenuated virus vaccines because these viruses do replicate in the body. As a result, it produces both cellular as well as humoral responses. So this would work more like a natural infection. But if you have an inactivated whole virus or subunit vaccines, they cannot replicate in the host cell. So as a result, they have a relatively less efficient induction of the immunity. So that's why we need stronger adjuvants, okay? So what can we use for adjuvants? We can use mineral compounds. So aluminum, magnesium are commonly used. You can use a specific type of system that's been developed for that vaccine. You can have bacterial products, you can have proteases, you can have other complexes that stimulate the immune system. It can have a combination system where you basically have the vaccine and multiple different adjuvants. And you can also have things like emulsions. You can also have tensoactive agents, okay? So these will, for example, change gene expression, okay? And there has been extensive research on newer technology and the delivery systems. So you can see all these different types of delivery systems as well as the adjuvant systems as well, okay? So again, read through this and understand it, but I don't expect you to know all of them. And one example of research that involves better technology and strategies for delivery. I'll show you one example, but you can have things like aerosol delivery, eye drops, suppositories, troches, various foods where you can actually genetically modify the plants, okay? And one of the newer ones is the gene gun, okay, which is basically this kind of cartridge. You precipitate, let's say, DNA or RNA onto gold particles. Remember, gold particles are going to be inert, and it's not going to cause any issues in your cells. You load that into this cartridge or into this tubing, and you basically coat the tubing with these gold particles. You insert it into cartridges, goes in this kind of gun, okay? And basically, you press against the skin and press the button, and it shoots the gold particles right under your skin, okay? So this will actually inject the gold particles, and then from there on, because it's coated with your DNA, a lot of this will go inside the cell because the gold particles will be able to pierce the cell as well. And once the DNA is in, you can start making proteins, and you can start making antibodies against those proteins, okay? So this is very Star Trek-like, where they basically, instead of injections, they use this kind of device to inject people, okay? Okay, and one thing to add on here is that a lot of people have hesitation about the vaccines, especially these days. But as far as we know, the current vaccines are the safest that are ever marketed. But as we know, nothing can be 100% safe, because even, for example, you drink too much water, it's not safe for you, okay? So there has been several unfortunate incidents for history of vaccine development. And this was partly due to incomplete knowledge on many of the immunological reactions. And people didn't understand exactly what was happening, okay? And most adverse events were very poorly understood or difficult to foresee under these conditions. And from there on, we developed worldwide surveillance initiative to make sure that the vaccines are safe as well as effective to bring up public confidence in these vaccines. So again, here's the vaccines, and here you can see some of the adverse events that have happened in the past, okay? So for example, the smallpox vaccines, one of the smallpox vaccines, once it was injected, it caused sepsis because it was contaminated with bacteria. Okay, so this was one of the earlier issues with smallpox vaccines. Okay, yellow fever vaccine. In this case, the lot was contaminated hepatitis B virus. Okay, so when people were given this vaccine, they all became infected with hepatitis B. Okay, so you can see that because of poor regulation and poor understanding of material, there were some major issues with the vaccine development. Because what we see is that vaccines create a dead end for the virus by blocking virus replication, as well as virus spread in multiple different ways. You can limit mutations, so it's harder to generate the virus variants if infection is not successful. You can reduce the viral load and transmission. So if you don't get enough virus from someone, you're not going to get infected. Because remember, a lot of these infections that are passed on, they are dose dependent. okay? So this will reduce the virus load. It will protect other people that have underlying conditions or even people that are unvaccinated. So this is where herd immunity comes in. This primes our immune system for reinfection, and if we do have reinfection, the disease is it's much less severe, okay? And it can lead to global eradication and protection. So you take those vaccines, and if everyone is vaccinated, that host will become dead-end host. So what that means is they won't pass it on to other people, okay? As a result, the infection stops, okay? Everyone clear on that? Okay, let's take 10 minutes and then we continue. Just to add on, something I'm going to show you before I let you go. We'll talk about herd immunity. And in one of the final exams, there was a question about herd immunity. And someone said herd immunity is when we vaccinate cows. We all had a good laugh about that. All right, 10 minutes.